Hello, welcome back to MLSQR, and we'll discuss immunology basics with the complement system for the clinical lab. Before we dive into the details, let's look at a broad overview of how complement plays an important function to fight infection and mediate inflammation. Later, we'll discuss details of the complement cascade with activation by the classical, lectin, and alternative pathways, with specific details about important complement proteins and some complement testing. The complement system plays a very important role as one of the key elements of the innate immune system. Barrier surfaces in our skin and mucosa of our oropharynx, respiratory, gastrointestinal, and urogenital systems provide a protective external defense against foreign invaders. But once this external line of defense is breached, a second line of defense of our innate immune system relies upon soluble and cell bound plasma proteins and immune cells to greet the foreign invaders. Complement is a complex series of more than 50 proteins of the innate immune system that responds rapidly and rather nonspecifically to infection and inflammation. Complement can lyse foreign cells and microbes, increase vascular permeability, recruit monocytes, macrophages, and neutrophils to the battle site, opsonize and tag foreign microbes to make it easier and quicker for phagocytosis, and help amplify the immune response. In our more developed immune system, activated complement proteins amplify the immune response and interplay with the adaptive immune system in a very complex cooperative effort. Activated complement interacts with many innate immune cells. Complement is intricately involved with inflammation. Unfortunately, complement plays a significant role in many aspects of pathology when overactive, unregulated, or self inflicted autoimmune inflammation damages healthy cells and tissues. Tissue necrosis, sepsis, drug side effects, transfusion related pathology, also any disease that can lead to a cytokine storm can initiate the complement cascade that exacerbates inflammation and hemolysis of red blood cells. There are 11 main complement proteins C1 through C9 if you count C1QRS as three proteins for the recognition unit. Initially, they are zymogens, an inactive enzyme form that requires activation to form an active enzyme. The complement cascade involves three pathways known as the classical, lectin, and alternative pathways. An enzyme cascade works in sequence with amplification, such as that seen in the coagulation cascade, and in clinical chemistry, we've seen linked enzyme reactions. C1q is the recognition unit in the classical complement pathway, combining with subunits R and S to form the C1QRS recognition unit. The function of the C1 is to bind to the Fc portion of the antigen antibody complexes, while C1s and C1r are serine proteases to activate the cascade. The classical complement proteins in our innate immune system complements the adaptive immune system, where antibodies secreted by activated B cells and plasma cells attack a foreign invader with foreign antigens and are recognized by C1QRS recognition units attaching to the Fc portion of the two antibody complexes. The term complement fixation means that immune complexes fix complement the C1QRS at the Fc portion, and these antibody complexes are IgM, IgG1, IgG2, and IgG3. Complement recognition is quickly followed by complement activation of C4 to C4a and C2 to C2b. The nomenclature refers to lowercase a and b as activated complement proteins with anaphylatoxin and binding functions in the cascade. C4b2A will catalyze the cleavage of C3 to form C3A and C3b, which binds to C4b2A to form C4b2A3b. Note the significance of C3b as we will see next. C4b2A is also known as C3 convertase. It is an enzyme that will convert C3 to C3A and C3b, forming C4b2A3b, also known as C5 convertase. Do you see patterns? C3 plays a major central role in all three complement pathways. It serves as the pivotal point of convergence, a powerful amplification, and represents the most significant step in this entire cascade. C3 is cleaved into C3A, an anaphylatoxin, and C3b, which binds to C3 convertase to cleave C5 into C5A, another anaphylatoxin, and also it forms C5b. Next, we will see the significance of C5b. C5b attaches to the target membrane, forming the first part of the membrane attack unit. When C5b binds to the target cell, it activates the cleavage and assembly of C6 to C9. Note the key concept that activated C5b binding to C6 through C9 to form the membrane attack unit, otherwise known as the membrane attack complex or MAC. The membrane attack complex opens a pore that allows an influx of water and efflux of microbial electrolytes, causing hemolysis and microbial death. In a similar fashion, red blood cells that are sensitized by antibodies attacking the red blood cell antigens, thereby activating the complement cascade, will lead to the membrane attack complex affecting hemolysis seen in pathology and the complement fixation tests, such as the CH50 test, which we will see later. Now that we are familiar with the classical complement pathway, let's look at complement activation by lectin. For your information only, lectin activation bypasses the antigen antibody activation in the classical pathway by direct recognition of surface molecules on pathogens known as lectins, picolins, and collectins. Some microorganisms have mannose sugars on their glycoprotein surfaces, which are recognized by mannose binding proteins. So, this is the lectin pathway of complement activation. Finally, we get to the alternative complement pathway, which also bypasses the recognition phase. One key note is that the most likely biomolecules to activate the alternative pathway is the one found ubiquitously in gram negative bacteria, which are the lipopolysaccharides. Factor B and factor D are involved in this pathway. Gram-negative bacteria with lipopolysaccharides on their outer membrane activates the alternative pathway beginning with C3. C3 is cleaved to form C3A and C3b, and factor D activates factor B. Activated factor B joins with C3b at the target microbe surface, where properdin stabilizes the complex. One ASCP study guide question refers to activation of the alternative pathway by properdin factor B. Another interesting note is that antigen antibody complexes containing IgA and IgE may initiate activation, as noted in another ACP study guide question. For individual components of the complement pathway, C3A has anaphylatoxin activity, releasing histamine and other mediators from basophils. A key C3b function is opsonization, coating the bacteria, making it easier for the phagocytes to phagocytize complement-coated bacteria. C5a functions as a powerful anaphylatoxin and chemotoxin, all of these contributing to the complement cascade, taking part in many bacterial killing mechanisms. For clinical lab testing for complement, there was one ACP study guide question about CH50. CH50 is a measure of total complement activity. CH50 is measured at 50% hemolysis at serial dilutions and expressed as a titer converted to CH50 units. For information only, there is a AH50, which is the alternative pathway activation hemolytic titration assay. C3 and C4 measurement by enzyme-linked immunoassay, there was two ACP study guide questions where C3 decreases in systemic lupus erythematosus because of the C3 nephritic factor linked to deposition of immune complexes in the nephrons. Also, C3 and C4 are most likely to coat cells. In the blood bank, we have a direct antiglobulin test, DAT, or direct Coombs test, which is useful for autoimmune hemolytic anemia, suspected hemolytic transfusion reaction workup, and cord blood testing for newborns. Direct antiglobulin test reagents include monoclonal anti-C3b, C3d antisera, and complement control cells. We hope to cover more complement testing in future videos in more detail. This concludes our introductory presentation on complement for the clinical lab. Stay tuned for our next video for the question and answer session that revolves around the ACP study guide for CLS, MLS, and MLT candidates for the licensing exams. Thank you for your time, attention, and support, and we'll see you in the next video.